everyone. So today we're going to be talking about chromosomes and the cell cycle. So we learned a bit about DNA and how DNA reproduces. Now we're going to learn about why DNA reproduces, right? We're making copies of DNA so that the cell can eventually reproduce. So let's see how that happens today. Before we even get into that, I just want to get into some fun facts with you about how much DNA is actually inside of a cell. So if you stretch out the DNA from just one cell, it's actually about 2 meters long, and that's from one cell. We've got trillions of cells. So if you put all the DNA in all of your cells, unwound it, stretched it out, it would actually be twice the diameter of the solar system. And I bet none of you believe me, but it's true. So let's see exactly how we get so much DNA in a really small confined space. All right, so in eukaryotes, remember eukaryotic cells are plant animal cells that have DNA that's enclosed in a nucleus. So pretty much everything that's not bacteria. Bacteria have a much simpler version of this. They have a circular chromosome, a little bit different. We'll talk about that another day. We're focusing on eukaryotic cells today. So all eukaryotic cells store their genetic information as DNA, which is in the form of chromosomes that are located in the nucleus of the cell. Most organisms have between 10 and 50 chromosomes. We're going to have a focus on humans in this class. Humans have 46 chromosomes or 23 identical pairs. So we've got 23 chromosomes from your mom, 23 chromosomes from your dad. Put them together, you got 46. Now let's take a look at the structure of DNA. So we learned about this part here, this DNA double helix. We've got the nucleotides connected at the center of the DNA molecule. Those A's match with the T's, the C's match with the G's, all that stuff. But what's happening is this double helix is wrapped around these proteins that are purple here. These are called histones. So they're kind of wrapped around those. And then those are even wrapped around and coiled around each other even more. So it's a really tightly, well-organized little structure that they're creating here to create these supercoils, kind of like a 1980s telephone cord. And eventually all that coils together to make up this chromosome. So let's talk about some important vocabulary words when we're looking at chromosome structures, and it might help to actually draw this in your notes. So normally when you think of a chromosome, I think most people think of that X shape, but really the X shape is when the chromosome is replicated. So when the DNA is already replicated. Normally, the chromosome is just half of the X. So this is called a chromatid, when it's just half of the X, and this is an unreplicated chromosome. So normally it looks like this. The center of this molecule is called a centromere. Sounds like center, right? Centromere kind of holds all the pieces together in the middle. When the chromatid is replicated, so when DNA replication takes place, then we get this X shape, so we know it's replicated when it's an X shape, and each half of the X is called a sister chromatid. I don't know why they're not called brothers, they're called sisters. And they're identical sister chromatids because they are exact copies of one another. So this half of the X has the exact same genes as this half of the X, right? That's the whole point of DNA replication. We get exact copies of one another, okay? So let's look again. The replicated chromosomes are called sister chromatids. They're held together in the center by a centromere. And again, the sister chromatids are exact copies of one another. And this is actually what it looks like on an electron scanning microscope. So we can actually see all these little tiny pieces of DNA. Pretty cool. All right, so DNA replication we've already learned about, but let's just sum that up real quick again. So before the cell divides, the DNA must be copied or replicated. Okay, so each new cell can then get a complete set of DNA. right? So we start with one cell, we end up with two cells that are exactly the same because of this process, the DNA replication process. So as we get into the cell cycle, there's a couple phases that you're going to need to memorize. Wouldn't hurt to maybe draw an image that looks like this. Let me try to move myself out of the way here so you can see. All right, so the cell cycle is kind of like the life cycle of a cell, right? Humans have a life cycle. We're babies, then we're kids, then we're teenagers, then we're adults, then we're old, and then we die. Okay, cells have a similar life cycle. The majority of their life is spent in interphase. So that's this like whole section here that's kind of blue and pink. So the G1, S, and G2 is all part of interphase. This is like the cell's adult's life. When they're just functioning, doing their regular cell jobs, they're in interphase. M phase is just that period where the cell is dividing, when it's going through mitosis. Okay, so this phase here, kind of in gold. And then cytokinesis is just when it completely divides. All right, so all of this is interphase made up of G1, S, and G2, and I'll go into those in a second. And then only when it's dividing, just like when a human is reproducing, right? Only for a couple years in their adult life are they reproducing. The rest of their life, they're just doing their job, going about their daily tasks. Same thing here. So let's start with the first part of interphase. It's called G1. This is where the cell just grows, performs its normal functions, just doing its day-to-day thing. If it's a muscle cell, it's contracting. If it's a blood cell, it's carrying oxygen around the body, right? It's just doing its regular job. G1. You can think of this as the first growth phase. G0 is just kind of when a cell has exited the cell cycle. So it's only doing its job and it never divides. This is like an adult that never reproduces. So it doesn't divide. It's never going to synthesize its DNA and go through mitosis. It just does its regular job until it dies. Some examples would be like neurons, right? Our nerve cells don't really regenerate. That's why if someone's paralyzed or has brain damage, a lot of times that really isn't all that reversible because those cells don't divide. So we need to take special care of those cells. Okay, so let's get back into interphase. So after G1 phase, then S phase happens. S stands for synthesis phase. Synthesis means to make. So this is when we're making copies of DNA. So the replication phase. This is important because we need to have a complete new set of DNA in order for the cell to divide, right? So if we've got our single chromatids here, after we replicate these, now we've got replicated chromosomes that look like X's. Okay, so the chromosomes will look like this during G1 phase. They'll look like this after S phase. Then G2 phase is when the cell's getting ready for division. Okay, so it's preparing for division. It's making organelles like centrioles that are needed for division, which we'll talk about soon when we get to mitosis. So it's just getting ready to divide, getting big and fat and plump. And then M phase is where the cell is actually dividing. So it's broken down into two parts. So mitosis, which you probably learned about in middle school a little bit, is the division of the nucleus. Prophase, metaphase, anaphase, telophase. Cytokinesis is that final part of the phase where the cytoplasm is split. Okay, so first we have to divide the nucleus, then we divide the cytoplasm, and the final product is that we have two identical daughter cells. So these daughter cells are going to be exactly the same as each other and exactly the same as the original cell. And then once they divide, they'll enter interphase all over again. Start with that G1 growth phase, synthesize its own DNA, eventually divide as well. So let's sum up. So a summary of these four phases. Interphase is made up of G1, S, and G2, where the cell grows, synthesizes DNA, and then grows again. And then M phase is the mitotic phase. So this is when the cell divides its nucleus first and then eventually divides its cytoplasm. Okay, so let's take a second and practice some of these new vocabulary words. So let's see if we can label the parts of the cell here with the words in this word bank. So pause the video for a second and see if we can answer. All right, so let's go over these. So we've got our DNA here. That should have been easy. This is our DNA strand. The histones are those proteins that the DNA gets wrapped around. The sister chromatid is each of these sides. So number 3 would be sister chromatids, right? Because we've got 1, 2 sisters. And then number 2 says the entire X shape. So that's representing the full replicated chromosome here. Okay. Right, now let's talk about these phases of mitosis, what happens in each one. So remember, mitosis is the division of the nucleus, and it only occurs in eukaryotic cells. Prokaryotic cells have their own simplified version of this. So there's 4 mitotic phases, and we'll go into each of them individually. Prophase, metaphase, anaphase, telophase. PMAT or PMAT is a good way to memorize them. PMAT. Okay, and I also like, okay, so if we look here, these are actual microscope images. These blue things here that can look like little worms, these are the chromosomes. So one thing that I always remember from when I was in high school is this little finger representation. So if you go like this, right? Your fingers are representing chromosomes. This is prophase. In prophase, they're all together. Metaphase, they line up across the middle. Anaphase, they come apart. Telophase, we have 2 new nuclei. Prophase, metaphase, anaphase, telophase. That will help you remember what the chromosomes are doing at each phase. So let's start with prophase. So prophase, we've got our chromosomes here. The nuclear membrane is starting to disappear. The chromosomes are becoming visible if we were looking in a microscope. And the centrioles are forming spindle fibers. So these little T-shaped things here are the centrioles. These guys are the spindle fibers, and that's what's going to help to pull the chromosomes apart. This is what it looks like under a microscope. So you can kind of see that they're starting to look like little worms, those chromosomes. You can actually see them. And before prophase, this would kind of just be totally purple. All the DNA wouldn't be in 2 chromosomes yet. Metaphase, remember, metaphase, they're lining up across the middle. So the chromosomes are attaching to these spindle fibers, which line them up across the center of the cell. Looks like this if we're looking in a microscope. Got those dark purple chromosomes lining up. You can see the spindle fibers here that help them line up. Anaphase. The spindle fibers are pulling the sister chromatids apart, right? So each half of the X, remember the X is a replicated chromosome. So each half of the X gets pulled to separate sides of the cell. Okay, so these are exact copies of the same information. Looks like this under a microscope, right? Prophase, metaphase, anaphase. Those chromosomes are getting pulled apart. And finally, telophase, or some people say telophase, is when the sister chromatids have finally arrived at opposite poles of the cell and a new nuclear envelope can form around each set of chromosome. Okay, so we've got 2 new nuclei forming here. And you can see the cell membrane is starting to cleave. It's starting to separate. All right, take a second, see if you can identify what these cells are in. All right, so you might have guessed that all the ones that have this kind of purple, plain looking nucleus, these are all in interphase. So these cells aren't dividing, they're just living their regular cell life. B is when we can start to see the chromosomes. So they're becoming visible, they're starting to spread out, so the nuclear membrane is probably disappearing. So this cell is in prophase. This cell, C, they're starting to line up. So these are metaphase. And D and A are both prophase. Okay? Hope this was helpful and we'll practice more in class. See you next time.